And thanks you all for being here. Since we are a rather small audience, you can interrupt my talk anytime and ask questions. Okay. So today I will talk about the trachea transplant scandals of Paolo Macchiarini. All of you heard of him? Yeah? Okay. But probably half of my talk will be about general concerns on research integrity, which and the bad practices in academia, which also made this scandal possible. Because it's not just about medicine, it's also about bad science. So, okay, so a brief summary about who Paolo Macchiarini is. He is a thorax surgeon, and he was celebrated stem cell pioneer, and he used to be the director of the Careggi University Hospital in Florence. And he used to be also a professor at the Karolinska Institute in Stockholm, Sweden. He transplanted a number of patients with trachea replacement made of decellularized cadaveric organs, dead tissue basically, and plastic trachea made out of plastic, obviously. And they were seeded with patients' own bone marrow cells and declared to be regenerated living organs. So all of his trachea transplant patients are either dead or in permanent care. They're lucky survivors. Nobody was cured. Okay. So, how did it get so bad? Was it just, as many in academia would like to say, well, it's just because Macchiarini is a very naughty person and we can't avoid this. Well, once every 100 years you just have a criminal in a system and you can't do anything about it? Or was it really a system which was partially to blame? And this is, well, it's obviously Paolo Macchiarini here. And also all the universities and university hospitals where he used to work, it started in Hanover, in Germany, the Hanover Medical School. And he went to Barcelona, University Hospital Barcelona. From there he went to Florence, to Careggi, and he had a guest professorship at UCL, University Hospital in London. He became professor at Karolinska, and he also had his main lab in Russia, in Krasnodar, with the Kuban Medical State University. So, is it just him, or is it the problem with the system? And I'm saying that it's both. And, well, of course, there is a special character of Macchiarini who knew how to play the hype and the greed in biomedical research, and he knew what to do when it got too hot for him, when people were onto his deeds. He switched institutions. He went from one to the next. So when things got too hot in Barcelona, he went to Florence, and when he ran into trouble in Florence, he went to Sweden, and so on. So he was always charming the new hosts with promises of fame and money, and every university was impressed everywhere he went, even the rich ones like Karolinska, especially them. And the important bit about it, the scandal was not uncovered because the universities wanted to deal on it. It was uncovered because of journalists, because of a number of Swedish journalists who worked for television and newspapers were on this case, and they reported it, and there were a 3-part television documentary which they made, and this is how Macchiarini's game ended, basically. What the universities are doing, all the ones I'm listed here, they're busy covering it up quite actively. All of them, including the Karolinska Institute, which pretends to have investigated in full and punished everybody responsible. This is actually not true. What Karolinska is busy is damage control, just like everybody else, especially Careggi. So how could it get so bad? Now I'll give you a crash course, if you're interested, how to get away with bad science. And since it's a medical faculty, how to experiment on humans free from all regulations. So basically, you probably already know that if science is very biased towards hot topics like stem cells and regenerative medicine. It's extremely popular. And of course, you expect it to cure diseases which are probably not curable at all, like cancer, or create organs from the lab. where there's organ shortage or the organs are not transplantable. So these are promises which are expected from you. And of course, if you want to publish a paper, it has to be a simple mechanism, nothing complicated, not the story of 5 years of your research, but a very simple discovery, like you discovered a gene for autism, or you discovered a gene for schizophrenia, or whatever. Or you have to make a genius correlation, which nobody would consider to make any sense. But because you work on a hot topic like gut microbiome, which is very, very hyped field these days, you should connect it to a disease which makes absolutely no sense. And then you get a paper in Nature. Recently, Nature published a paper claiming that the gut microbiome of a pregnant woman causes autism in her children. It makes absolutely no sense scientifically, but this is published in Nature, and this is it became truth because Nature has an impact factor of 43, and it means everything there is true, right? So you make a correlation like this, you get into Nature, you get into all media. And of course, everything you do must be translational or commercially applicable, especially if you want to apply for EU funding, because the European Union is not interested in funding science in reality. They want to fund something which is marketable. And this is why the European Union funded 2 trachea transplant trials, one by Macchiarini, which they terminated, not because people were dead, but because Macchiarini switched the model to something which was not patentable and had no novelty in it, the trachea plastic material. And so the European Union said, well, we can't market it, so we're terminating the grant. They never cared about his dead patients. And this is why the European Union is funding another clinical trial, which I will tell you about later, by Macchiarini's collaborator, using exactly the same model, because the European Union thinks there will be a market leader in trachea transplants, and the Italian university is part of it, and I think it was Pisa or Bologna, I'm sorry I forgot it now, but it's mainly focused in London, and also the University of Munich is part of it. They're going to make tracheas there. So this is the bias. So no more general, why do scientists manipulate data? And I believe there are 2 main reasons for this. The cartoons are mine, by the way. If you want to use them, free to do. I will post the talk online. So the reason one is to prove a preconceived theory against lack of experimental evidence. Because a lot of scientists start a research project, they already know the result. They already know what their paper will look like. So your job, as for example, as a PhD students, would be just to fill in the blanks, to provide the right figures. And sometimes it obviously doesn't work, so you have to fake the data or manipulate it a bit. So as a result, of course, your paper is not reproducible. You polluted scientific literature, especially if it's in a Nature paper. Then everybody has basically to confirm you. Every paper which trying to disprove you will not be published at all. You suffocate people who have the correct models, and you control the research field. So when you're caught, of course, it's a huge damage to your entire research field. But then it can be even worse, because there's a second reason to fake data. And this is to scoop somebody, to scoop a competitor. Imagine you went to a very big conference and you went to a poster session and you saw something very interesting, a very cool result, which on the same field as you work, and it's not yet published. So, right? Well, you can go back to your lab, to your computer, Photoshop some gels, and publish a paper very quickly, right? And then you scoop this person. This is how science works. The person who published first is a discoverer. Everybody else, well, nobody cares, right? So the original discoverers will have difficulties to publish their paper. Obviously, the outcome is the same. You control the research field. You get all the funding dishonestly. But this is the tricky bit. When you're caught, what happens? When people discover manipulations in their paper, well, you can say, everybody says, yes, but the results are reproducible, right? Right? Others reproduce it because you just take genius. Yes, you manipulated data, but it doesn't matter because the result is true, right? So nobody will be angry at you in the end. So I know such cases, in fact. So I made also 3 categories how scientists helped a data to fit the theoretical model. So what is very common is the selective data acquisition and omission of critical controls. I'll talk about it in detail. Adjustment or manipulation of data, which is less common. This is heavy manipulation. And what is very, very rare is actually the delta falsification fraud. But every time you talk about research integrity, they will be talking about these rare cases, like Diederik Stapel, you know? Have you heard of him? The psychologist. He's the only fraudster in the history of science, officially. And the reason is because he admitted it. When they found him out, he just said, yes, I invented it all. He was sacked, of course, by his university. He started to write books about about his faking experience. So Diederik Stapel is basically the only fraudster in the history of science, because nobody else admitted it, obviously. Yes. Dutch. Well, because he's the only one. You can't say it, because all the other scientists, nobody in the US, Too much, right? Yeah. Yeah, well, he's a special case. That's why everybody talks about him, because he admitted it. And otherwise, well, I don't know if you can think of some cases, but this is a problem. This research misconduct is always presented as something so rare that you shouldn't bother about it. It could never happen to you, right? You are not Diederik Stapel, right? But this is exactly which is not true. So this is the first circle of hell, let's say, put it like this, and you will recognize that it's quite common, right? You make small sample size or do single experiments, right? You keep your experiment so small that your result becomes significant because you use too many, just 3 mice, or you did the experiment only once and never repeated it, right? And you publish this. So cherry picking, you remove the odd data, the outliers, which you don't like, right? So you get a nice result, so you get your difference, for example. Control avoidance, for example, if you know which control you have to do, and you don't do it. For example, you get a knockout mouse model from somebody, right? And you just trust that it's a knockout mouse model. You never actually test if the protein is still there and if it's functional, because it's written on the mouse that it's a knockout or in a cell line, right? So you trust it. You don't do these controls. And then you deliberate artifacts. It's also very popular in science, where you just stick and hoard an obscure reagent because it's the only one producing results. And this is, you know, you have a single batch of antibody which produces a certain signal, yeah, and you only use this batch and you buy all of them if you can afford it, right? Because you like the result. Because does it make sense that every other batch of the same antibody, every other manufacturer does not produce something else? Maybe it's your batch which is faulty, right? This would be a reasonable explanation. Maybe your batch is recognizes something wrong. No. Because you like the result, you keep sticking to it, and you proudly say, well, it's reproducing now because we bought all the antibody, or the same with primers can happen, right? You so you recognize these things. This is more like a second level for more professional ones. Heavy cherry picking, so you really start removing entire data sets because they are wrong. Half of the data set can go because you decide it doesn't fit. Very popular in clinical trials. When you start removing patients, because they didn't improve after your treatment as you wanted them to. You say they were wrong patients. Or you start changing baselines, for example, that you started to try with one baseline and then you decided, oh, we'll change it. So then suddenly it becomes significant, right? Very popular. Triplicating. There are statistical tools to turn a single experiment into a triplicate with error bars. You didn't even have to invent data. You can do it, and then everybody will think you did it twice. P-hacking is very popular in psychology, but also in clinical trials. So if you know the scientist John Carlyle, an anesthesiologist, who is also a statistics specialist, because of his statistics skills, he caught a fraudster in Japan. And he also published recently a paper where he showed that a number of papers in the big journals have very suspicious statistics. So definitely worth looking at, which with baselines, actually. He noticed their baselines are statistically a bit fishy. So this is quite popular. To p-hack until you get your significant result. And of course, it has to be below 0.05, right? This is why somebody even analyzed and turned out that most scientific results are exactly just below 0.05 for some reason. And loading controls. You know, everybody of you who did have a Western blot or a different gel, that you load your samples, right, to see a difference between samples. But you, of course, need a loading control. You need to see that you loaded the same amount, right? But this is where you can start tricking. You can run two gels. One gel is to measure the difference, and one gel is to do a loading control. And it's not like the only students do it. Even PIs teach students to do it because they say, well, those are the same samples. It's okay, and you load the same amounts even. What is not accounted of, nobody knows how much is actually loaded. You know, only the person who is holding the pipette knows how much was loaded in every single well, right? And in the end, you have mismatched loading controls to the analytic gel, and it becomes useless, of course. But this is how you can make a result. You swap loading control gels between gels or make libraries. Do all possible other tricks. With real-time PCR, you know that the result is an Excel sheet, right? One can do whatever you want there. So those are the methods. And then scientists say, but those are just controls. They don't matter, right? So, but this is the most important part of the Western blot, the loading control. So, and the third part is obviously the falsification and fraud, which I mentioned, which is where people really go and say that, well, I need to publish this paper quickly, or I need to convince a peer reviewer, and I don't have the time for it, so I'll just fake it because I know how the experiment works out, right? Or the fraud where you really have enough of biology or medicine, and they think they're stupid, and they don't get your grand reasoning, so okay, if the experiments don't work as per, I have to make them. So it happens. So people fake data to get the theory published. Once it's published, it's like carved in stone. It's like the 10 commandments by Moses. Nobody can dispute them because it's a public evidence of success. It was peer-reviewed. It means it's trustworthy. The higher the impact factor of the journal, the better the peer review. This is a saying. So if it's in Nature, it has the best peer review ever, and that's why it's absolute truth. Paper in Nature Communications is probably half as true as a Nature paper. I don't know how it's measured. Half as reliable, I don't know. But you can't dispute a Nature paper, for example. So the content does not matter. Scientists never congratulate each other on what they publish. Nobody reads each other's paper. You say, a very nice paper in Nature Medicine you had, right? No idea what it's about, right? or in EMBO journal, right? This is how it goes. So the paper and where it's published counts. And you never criticize the published paper in public. Never. This is the worst thing you can do in science. It is the rudest thing it is. You know, it's more polite to walk up to a professor and kick him in the testicles than to criticize his paper in public. And why it's so? Because science has a climate of fear and a coalition of silence. And this happens because science is a situation where simultaneously cooperative, competitive. You have to cooperate with people you're competing against. And of course, science is depending on funding. In Italy, you all know the problem, right? That you can't afford anything. And so you know when you have to shut up, be silent, and just accept everything, because the person who runs the funding program, or person who will probably be your peer reviewer, well, even if it's a worst fraudster, you know, you have to be silent and be polite. So that's why scientists rarely dispute each other publicly and report misconduct. This is also what happened in Macchiarini case. And now, of course, this is as an aside, scientists do talk about misconduct, but most of the case to damage a competitor. And many cases, this is a false misconduct accusation. For example, if you you crossed your boss, and he wants to destroy your career, he or she, they will start telling everybody that you fake data on telephone. You will never know that they did it, but you will notice that nobody invites you for an interview. So it happens in science to damage competitors, critics, rogue ex-employers. I know that people lost their jobs like this for messing with fraudsters, actually. So back to Macchiarini scandal finally. It had everything. Obviously, he came in the right time when the hype for stem cells and regenerative medicine was the biggest, where especially Sweden was investing enormous amounts of money in stem cell research. That's how he ended up in Karolinska. And he published in one of the most respected journals in medicine, The Lancet. He drew huge funding, and he had political support, obviously, especially in Italy, is an issue. You recognize this is Enrico Rossi, president of Toscana. And Enrico Rossi still defends Macchiarini. He still says that Macchiarini is a genius, and he said, well, if we kept him in Florence, we would control him, and no bad things would happen, and we would have the best surgeon on the planet, something like that. So they're still friends, obviously. So maybe he'll come back. You never know, right? So obviously, Macchiarini's science is, to be polite, is extremely simplistic, and is absolutely not supported by any scientific evidence. It's just wishful thinking. Science fiction, basically. Macchiarini misrepresented his research results. The original data was lost or never existed. He misrepresented clinical outcomes by presenting very sick or dead patients as alive, and he manipulated data, of course, also. He committed ethics breach at every step. He was constantly lying about getting ethics approval, which he never did. The institutions have threatened the whistleblowers, especially in Karolinska. The 4 whistleblowers who reported him were threatened by the police and with police investigation by sacking by the Karolinska Institute. This is how Karolinska responded when they blew the whistle on Macchiarini. They threatened them with sacking. Of course, the investigation was suppressed. Maybe you heard that Karolinska did the first investigation and declared that Macchiarini did nothing wrong whatsoever. And in Careggi, nobody just investigates, so they don't have to invent anything. They just don't investigate. 5 people were transplanted in Careggi. And legal attacks on journalists. This is my case. Macchiarini's partner is suing me in court in Germany all the time. So now we'll show you how to get there, how to transplant trachea. Okay. So, why trachea? Trachea is an organ which you basically cannot transplant. I'll teach you why. Tell you why. So it is obviously a living tissue. It consists of cartilage rings on the outside. On the inside is a ciliated epithelium which moves all the particles out of the airways, bacteria, viruses, everything. So that's why. And of course, obviously, it's vascularized. Otherwise, it wouldn't be a living tissue, right? It depended on blood supply. A lot of patients would profit from a trachea transplant because they have damaged trachea or cancer of the trachea. But the problem is this organ cannot be transplanted exactly because of the way it is vascularized. Trachea receives blood supply from very tiny blood vessels, and a lot of them, and you cannot surgically connect them all in time. So with the open chest of the patient, you cannot do it physically. That's why it is simply not transplantable. Same is basically also for esophagus. It's unlike with lung or liver, where you have a central vein and central artery, you just attach those and then it works. No, with trachea it doesn't work. So if you do not connect the blood supply, obviously the new organ will die. So how Macchiarini and his partners circumvented this? They said, we will grow patients' own trachea, so you don't need a transplant, no immunosuppressive drugs, from patients' own cells on some dead scaffold, and say it is alive. Then we transplant it, and of course it has to be vascularized still, right? And then they said we found a magic method to stimulate vascularization. So the blood vessels will grow with a speed which was never measured in history of science. And then they publish papers, collect funding, awards, fame, promotions. Problems arise when the critics start come out and patients start dying. You have to silence it all, and you go back to point 3, publishing more papers and so on. So here's a bit to another practical consideration. So you need to grow a trachea. You said I will grow a trachea, right, with patients' own cells. You cannot use IPS model, the induced pluripotency, if you heard of it, because the technology is not developed yet, and there's a cancer threat anyway. So what you can do, you can take any cell type of patients which you happen to be working with and say, well, I think this cell is pluripotent and can generate any kind of tissues, and I will use that. And this is what they did. And it was not Macchiarini who claimed bone marrow is pluripotent, it was Nature paper. So that's why it became truth. It was a Nature paper published by the Belgian scientist Catherine Verfaay in 2002, where she claimed that the cells in the bone marrow are pluripotent. And people spoke of Nobel Prize, and she's on a stamp. She was in all the media. She was celebrated everywhere. But sometime later, scandal came. She had to retract a related paper because it was manipulated. And in her Nature paper also proved to be manipulated. But she didn't retract that. It was corrected. And when you avoid a retract and just correct a paper, well, the paper still remains true, right? And so, even the scientists proved since then that there is absolutely no pluripotency in bone marrow cells. The idea is still out. People still publish papers on it. In reality, nobody knows what this non-hematopoietic bone marrow cells do, so mesenchymal cells, not the ones which generate blood cells. So people have different models, but no sane person would say that they are pluripotent. So the next person, so this is Anthony Hollander. Now we're coming back to Macchiarini. He's a collaborator of his. He used to be in the University of Bristol, and he claimed that he can use bone marrow cells to generate cartilage. And this is what from the University of Liverpool, where he is now. It's written, 2008, Professor Hollander and a team of scientists and surgeons successfully created and transplanted the first tissue injury trachea using a patient's own stem cells. The bit about successful is completely wrong. Even Anthony Hollander knows it by now. He would never say it was a success, but they're still claiming it, and he has also a company marketing it. So this is the trachea transplant team. Anthony Hollander was the scientist, the basic scientist who provided the technology. He was not basically part of the transplant team. Obviously, you have here Paolo Macchiarini with the patient Claudia Castillo. She had tuberculosis of a lung, and she has a constricted bronchus, not trachea. She never received a trachea transplant, as it was celebrated, but a transplant of the bronchus. You all know how trachea looks like as a Y. So if she received a trachea transplant, she would be dead. But with the bronchus, you can still use the other lung, right? So the other lung was functional, that's why she survived. By now, she lost the lung. It had to be removed after, and the graft probably was also removed. So then you have Macchiarini's partner, Martin Burchell, who also used to be in the University of Bristol at that time. And he's a laryngologist, but at that time he was leading a veterinary lab, which is, of course, very sad for a clinician. But this is where he did research on pigs. And this is where he prepared the organ for Claudia Castillo in a veterinary lab. You can imagine how illegal it is. This is why he never reported it to authorities. And I found it out and reported, but still, so University of Bristol just told me that they don't, so the records on this experiment do not exist. They disappeared. Imagine that they're just 9 years old and everything was destroyed apparently. And this is Philipp Jungeblut next to Macchiarini. He is his student. He did medical degree under Macchiarini at Hannover in Germany. He's German over there. And he followed him everywhere, to Barcelona, to Florence, everywhere. And he's the one who's suing me at court all the time because he wants to be recognized as a successful scientist who published in The Lancet with Macchiarini and so on. But he also wants the public to know that he actually never had anything at all to do with Macchiarini's trachea transplants. So he wants both. He wants the fame for the science of trachea transplant, but he never wants to be associated with them. Oh, well, the German judges, they kind of follow this logic because he's a doctor, right? For now. Let's see. Now, how the principle it works. So Those slides are a scheme was made by Pierre Delare, who is a trachea surgeon from Belgium, and very vocal critic of Macchiarini and his method. So you take a dead scaffold, piece of plastic, or a cell-free carcass of a donor trachea. You seed it with bone marrow cells, with or without bioreactor, and you just stick it in. You cut out patient's trachea and you put it in. And then you just say it will start growing, vascularized immediately, and then you have a living trachea. The point is you can't save those patients. Once you remove their native trachea, they can't be saved anymore. So, yeah. This is Claudius Castillo's trachea in its bioreactor in Bristol. There was a press release photo, and they removed it, of course, the University of Bristol, but I saved a copy. And there you see Martin Burchell holding a plastic trachea from UCL, which he transplanted. And his partner is Alexander Seifalian, is a nanotechnologist who made this plastic carcass. So when UCL was investigating the trachea transplant scandal, they did, of course, they sacked the person responsible for the transplant, Seifalian. They said he will. Burchell remained in his position. UCL pays him £500,000 a year on salary. So whenever people found criticism, the answer was basically magic. So when people ask, why would bone marrow cells form cartilage around trachea? Because it's magic. And let's assume they formed cartilage. But how can they survive without blood supply? Everybody was asking Macchiarini this. Oh, he said, well, I developed a magic method where you add erythropoietin to the patient, and then suddenly the blood vessels grow out of nowhere. And it was accepted. And then, how can plastic become living tissue? Because Macchiarini was saying that plastic tracheas come alive. And people said, how can plastic be living tissue? Well, Macchiarini said, because it's magic. It's nanotechnology, right? Nanotechnology is different. It can do things. The danger of plastic in a material in a trachea is trachea is a moving organ because it's inside the chest, as you probably know. And if you have a tube plastic, it will start moving and it will tear apart all the organs around it. And this is what happened to the patients. It tore apart. Everybody had a hole in the esophagus immediately because the plastic trachea was tearing into it. And they started to bleed internally, and eventually they died. But Macchiarini knew, of course, what will happen when you put plastic. But he said, but this won't be plastic anymore. It will be living tissue, right? So can you imagine this logic? Some practical consideration. This is also what Macchiarini was thinking. He had a problem with bioreactors. I told you they made the first trachea for Claudio Castillo in a pig lab, in a veterinary lab, illegally, behind the back of authorities. And apparently he didn't want this to happen again, because this is illegal. So he said, human body is the best bioreactor. We don't need a bioreactor. We just put a dead, cell-free, cell-stripped carcass of a donor trachea into a patient, paint cells on it, put some erythropoietin, close it up, and it will grow itself. This is what he did for, 8 more patients. And then, of course, there's another problem, that if you use cadaveric trachea, you need a donor with a permit, you need time to decellularize it, and you need a GMP lab. And this is a serious problem if you want to be very quick and transplant people on as fast as Macchiarini wanted to. So he decided to switch to plastic, because you can make a plastic trachea in half a day. Custom made even. And this is the first patient. This is Ande Marian Bayen. He was declared dying. He was operated in Karolinska Institute. He came to Karolinska to be examined by Macchiarini because he had a slow-growing form of cancer. Macchiarini suggested, well, I will transplant you with a new trachea, and suddenly they did it. He talked the patient into this, and the patient died, obviously, and he suffered quite a lot. So the outcome of trachea transplant, Macchiarini transplanted 9 patients with plastic trachea. 8 of them are dead. 1 survived because the transplant could be removed. 10 patients received a cadaveric trachea from Macchiarini and or Burchell. So those few who are still alive are not cured. They are alive despite the transplant, not because of it. Nobody was helped. This is another, sorry, I put up the wrong paper here. But this is, I want to actually put another different Lancet paper, where this boy was transplanted, Kieran Lynch. He's still being paraded around as an example that the method works, by Martin Burchell, who runs the clinical trials now. But the problem with this boy was he never had a functional trachea. He was born without a trachea. He had a homograft for 10 years, which functioned very well, and then replaced it with a new model. Macchiarini put a new trachea for it. They had a fight, Burchell and Macchiarini, so he kicked him off the paper. I'm sorry I put up the wrong paper here. So, again, to more practical consideration, if you develop the technology of organ transplant, of regenerated medicine organ, you obviously need to test it, right? And here you can test it either on a pig, which is a standard model for human organ transplant, or you can test it on a patient. I'll give you the, so I put them side by side, and you will see that it's completely stupid to test it on pigs. It's so impractical. Pigs are large animals, right? They cost a lot. You need ethics approval. You need to explain why you need to do it. And the money is a huge issue with pigs. You obviously need proper controls. You can't just operate one pig. You need a control pig for standard treatment, untreated pig, and so on. You need to make survival rates. You need to report all the side effects if you want to publish it. You cannot just, if you operated 12 pigs, you cannot just find one which is still alive, publish it, and say the method is a success. Every reviewer will tell what happened to the other 11, right? Okay? Media is not interested. They don't care animal expenses. And, of course, others can reproduce your result. They can get their own pigs and try your experiment. So this is why pigs are very impractical. Patients. First of all, you have no costs. Actually, you will get paid, because the health insurance paying for this, for your experimenting, right? You do not need any ethics and regulatory approvals if you get a hospital exemption. A hospital exemption, an informal hospital exemption, is a permission given to you by your hospital directors to test a novel method on the patients. Then you get this agreement, you go to the patient with a piece of paper saying, sign here. Doesn't matter what you write on this piece of paper because nobody will see it, only the patient. And the patient, if the patient dies, won't complain anyway, right? So it's called informed consent, but most of the time, in many times it's misinformed consent. You don't need control, so obviously you can't get a control patient to operate, right? You don't need to compare with, because it's a single case experiment, right? If the patient is dead or is mutilated, you don't have to report about them. There is no law, like with clinical trials, which mandates that you must report the outcome. With hospital exemptions, you don't have to report anything. The patient ceased to exist. There was never such a patient. Right? If somebody asks, who is this? You don't remember the name, right? You never mention this patient. You only talk about what you think is good enough to be published. The alleged successes. And of course, nobody can reproduce it because you got the hospital exemption they want. And they won't even do it because it's unethical, right? But it's perfectly legal. So, of course, and then, of course, you should transplant only organs which you can't see from outside, like a trachea is. You shouldn't go and transplant arms and legs and fingers, right? But if you think this is silly, but there is a scientist called Harold Ott in the US who is claiming he can make every single organ, but the same method as Macchiarini does. He takes an organ from a donor animal, obviously. He removes all the cells of it, and then he puts bone marrow cells on it. And then the scientific argument is the bone marrow cells, they look around themselves and see which shape they landed on. Does it look like a heart or a bladder or a lung? I'm not making it up, seriously. And then they grow this organ because they see the shape. Yes. So this is how he claimed that he can regenerate a leg of a rat. He said, well, it was for bilimb, rat, forelimb. What he did, he stripped a leg, killed the rat, cut off its leg, stripped all the cells, put bone marrow cells on it, said, I regenerated it. All the media reported. Nobody was critical. Not a single critical. Nobody laughed at it even. Of course, what he did, the clever bit, he didn't stick it on a rat, of course, because then the people would start laughing, right? So you need to know where to stop, where not to make yourself ridiculous. And this is Pierre de Lare showed the same with the thumb. So basically, imagine you do the same with the thumb, right? You would just see a rotten piece of, not even flesh, right? But this is exactly what Macchiarini's trachea look like if you look inside with an endoscope. You have to imagine that somebody did it with a thumb. Exactly, the tracheas looked exactly the same, but you can't see it because they're inside a patient, right? So patient gets stents and tracheostomies and everything to keep them alive because the trachea was rotting. And then you have to know how to create a hospital exemption. You have to declare medical emergency. Sometimes you don't even have to say the patient is dying. You just say, well, the patient is incurable. So let's try something else. So you can skip everything, animal test approvals, everything with hospital exemption. Also with cancer patients, of course they will die eventually. The trachea cancer is lethal. So there are two types of trachea cancers. The squamosal carcinoma is very fast and lethal. This is what smokers get. But none of Macchiarini's patients had this cancer. They had a different. They had a very slow-growing form of cancer, which takes many years to metastasize into lungs. And there are standard treatments for these patients which can prolong their lives. But eventually, of course, the cancer will reach the lungs and kill them. Macchiarini said, if I transplant your trachea, you will live you will not die of cancer, which makes no sense, even if his method worked, because the cancer will still continue to metastasize. You cannot always remove all of the cancer. But the patients, of course, they believed him. Their parents believed him. They talked their children into this. And the scientists believed it as well. And another important tip, you have to publish quickly before your patients are dead and you can't publish it anymore. So you have to declare it as a success very fast. And your follow-up reports may become unrepresentable or in need of heavy beautification or lying, like in case of Macchiarini, where he published a 5-year follow-up on Claudio Castillo, which is just a bunch of lies. The important point, Macchiarini published 3 papers in Lancet, 2 on Claudio Castillo, 1 on Andemariam Bayen with plastic trachea. None of them was retracted because Richard Horton, the chief editor of Lancet, declared that Paolo Macchiarini is not guilty of scientific misconduct. He still stands by it. And you maybe heard of Lancet and Horton in a different context, in the Wakefield scandal, when Lancet published a paper by Andrew Wakefield claiming that vaccines cause autism. So Horton was worked basically in the same hospital ward as Wakefield. They were friends. And he defended him for 11 years and prevented a retraction of that paper. So whenever you have another measles epidemic or something else, thank Richard Horton, write Richard Horton a thank you letter, because this is his fault. He's still chief editor of the Lancet, and now he's teaching the world that plastic tracheas are safe. Okay. Next slide. Another point. So if your transplants don't work, like in Macchiarini's case it happened, you have to blame the patients, obviously. What he did, he said the patients were too weak. They had cancer. Cancer patients are bad. They're not strong enough to take the beautiful organ I created for them. I need healthy patients. Healthy patients means patients who were victims of accident and had a disrupted trachea and have a tracheostomy, an artificial hole which they breathe through. They are otherwise absolutely well. They can live get very old if of course they don't try to swim. So those are the 2, for example, patients. This is Yulia Tulik from Russia, who received a plastic trachea which killed her. And this is Yesim Chetty, a 20-year-old young woman from Turkey, who was a victim of a botched operation, which ended her with a damaged trachea. And her father heard of Macchiarini's miracle, and he brought his doctor to him. This woman suffered for 4 years after the operation. She received 2 plastic tracheas. Afterwards, she went to the US and received a transplant of lung, esophagus, and trachea combined. It didn't save her life. She died early this year. But this is a method where the doctor really says, I don't want very sick patients to test my method. And you have to imagine how sick it is. He's not talking about saving lives. He's saying, I want healthy volunteers, which I declare medical emergency. Obviously, you have a tracheostomy, you can't be helped. There is no method to cure tracheostomy. So he declared he operated those people under hospital exemption. Okay, so when Macchiarini was asked on Swedish television if he's a pathological liar, he couldn't say no. He couldn't even deny this question. It was very funny to hear this. So he insists that all patients were very sick, and he extended their lives, and everything is a big conspiracy against him. Again, 8 of 9 plastic trachea transplant patients are dead. He operated 9 patients with cadaveric trachea, 5 of them in Italy. And we only know them because the documents were found by Corriere Fiorentino journalists who found the documents proving the existence of these papers. That's how we know they existed. Otherwise, they would disappear. Nobody would have heard of them. Obviously, most are dead. This is one of them. This is Jadira Glikova, operated in Russia, in Moscow. It was a show operation. You know, they invited Macchiarini to Russia to save this young woman. Again, she had a traffic accident. She had a tracheostomy. She was absolutely fine otherwise. And he operated her, and now she's a very sick woman. When they say she is alive, but she is not doing very well. The transplant was removed long ago. She had several other operations because of this. But they're still celebrating her as a success. And this is Martin Burchell. He was a partner of Macchiarini, obviously. And on several cases, if you want to know details, go to my site, given his name, you will read a lot. And he's still a celebrated scientist. He's a poster from a British parliament where his achievements are celebrated in transplanting tracheas. So Burchell, obviously, he has an interest to distance himself from his past partner. And he and his partner, Martin Elliott, who is also in London, they say that everything was Macchiarini's fault. They had nothing to do with the bad things. In fact, Burchell says that he was always against using plastic trachea, always. He was always telling Macchiarini never to use plastic trachea. What actually happened is Burchell himself transplanted a plastic trachea with Macchiarini telling him not to do it in London, and it ended with the dead patients. But what happened afterwards, Burchell convinced every single science journalist in the world that trachea transplant, plastic trachea, was done by Macchiarini and not by him. That's how it works. So you create your artificial reality. And this is actually the patient. This is Keziah Shorten. She was Macchiarini's and Burchell's patient in London. Then somehow they decided to transplant her with a cadaveric trachea in Florence, where she was transplanted. She was sent back to UCL, to London, where they completely screwed up her post-operative treatment, which they tore up her esophagus in the process and so on. So Burchell then transplanted her with a plastic trachea, which is officially Macchiarini's transplant. So he was never near. And she died soon after. And this girl, she was 15-year-old Shona Davison. She received a cadaveric trachea transplant in 2012 from Burchell and his partner Martin Elliott in London. And this girl never exists because you cannot blame Macchiarini for her, really. Wherever you turn it around, you can't connect her to Macchiarini. This is why she stopped existing. There was a BBC documentary celebrating her magical cure. This BBC documentary is not available anywhere. The girl died right after the operation because what Burchell and Elliott did, they transplanted her with a trachea by giving false evidence to the Ethics Commission, and then they sent her away home right away. They knew that 8 patients transplanted the same method before were either dead or in terrible condition and needed a stent to be alive. They never put a stent in her. They sent her home. The trachea collapsed, the girl died. So they knew how the tracheas performed, but they wanted to prove that it will work in this case, but they wanted to prove it too close to them. So, and now they pretend they don't know how she died at all. Those are the doctors who transplanted her. So, you think it's all over, ancient history, and everybody learned their lesson? No. Everybody of you who pays taxes is currently funding a trachea transplant trial. Well, there is an INSPIRE trial, which is by Biologist. It's in UK, obviously it doesn't apply to you. But the follow-up, the phase 2 clinical trial, which is also run by Birchall, which EU funds for 7 million, is funded by your taxes. And the EU is determined to run it. They told me it's none of my business to ask about the state, any information about this trial, because it's a industrial investment, and so it's secret. The institutions which are involved mostly are in UK, but there is also Italy. Again, I think it's Bologna. Then in Munich, they're going to make tracheas, and they're going to transplant patients in Poland, in Italy, and in England, if they succeed. So basically, the idea is that in next year or in 2 years, everybody forgot about Macchiarini, will get their ethics approval, and you will never get to see it, and they will start transplanting patients in all over Europe. The company which runs this is called Videoregistrazione, it's based in Liverpool. Birchall is a paid advisor of Videoregistrazione, this is their boss, and this is how the Videoregistrazione presents the technology, how easy it works. You know, constricted trachea, you cut a bit out, put a new bit in, and it functions, like with cars. Yep. Well, of course, no. There's an important question. So, obviously, European Union grants are peer-reviewed, but you don't know by whom. You never see the reports. And I also know that European Union doesn't care what the reviewers write. If they want to fund the projects, they will. I have an evidence from the ERC grant, which was the reviewer pointed out manipulations in the paper, in the data underlying the project. And the European Union sort of, well, thank you very much for your review, but sometimes the reviewers' opinions do not align with the idea of the Commission. So this is how you will, if you ask them for peer review proposal, they'll tell you no, because it's commercially sensitive. Everything European Union funds is commercial sensitive. That's why you can never get any information, never know the reviews or who did it. Yeah. You have to trust them. Even the reviewers rejected it. It might get funded because they wanted to. Yeah. So, I think I'm almost finished. Yeah, I was actually. This is my last slide. Again, thank you very much. My website, my email if you want to reach me, and I'm always on Twitter if you want to follow me. Okay, thanks a lot, and hope you have questions.